Hello dear students, today we will discuss about the next topic from the chapter reproduction in lower and higher animals. Next topic is the gametogenesis. The gametogenesis, which is the process of formation of gametes, male gamete and the female gamete. So, the male gamete is the sperm and the female gamete is the egg or the ovum, and these gametes are formed from primordial germ cells of the gonads. First, we will discuss about the process of spermatogenesis. The process of formation of the male sperm or the male gamete, this is also known as the spermatozoa from the germinal epithelium of testis, is called spermatogenesis. At the onset of the puberty in males, the hypothalamus, it begins the secretion of the gonadotropin releasing hormone. In short, it is called the GnRH. This hormone, it initiates the significant increase in the secretion of follicle stimulating hormone, that is the FSH, which induces the spermatogenesis process. And each seminiferous tubules, it is lined by a single layer of this cuboidal epithelial cells. These are called the germinal epithelium. The germinal epithelium undergoes spermatogenesis, and in the process of spermatogenesis there is a production of the sperms. Process of this spermatogenesis, it involves the three phases. First phase is the multiplication phase, second phase is the growth phase, and third phase is the maturation phase. These are the three phases of this spermatogenesis. So, here is the diagram of this spermatogenesis process, and this is the first phase of this spermatogenesis, that is the multiplication phase, second phase is the growth phase, and third phase is the maturation phase. We will discuss about these three phases one by one. Multiplication phase. In this phase, what happens? The primordial germ cells, these are 2n in number, that is diploid in number, of seminiferous tubules, that undergoes the repeated mitotic divisions to produce a large number of spermatogonia, and the spermatogonia, these are also the diploid. Each spermatogonium is diploid and with 46 chromosomes. Now here mitosis and meiosis. Already you have studied about this process of mitosis and meiosis into the 11th standard. There is a separate chapter for this cell division. So if you have any doubts regarding this, you can check or you can read the information or the concepts about this mitosis and meiosis into the 11th standard textbook. Second phase, that is the growth phase. Some of the spermatogonia, they stop dividing and grow in size to develop into a primary oocytes. These are also diploid in number due to the accumulation of food. Then third phase, that is the maturation phase. So, in this phase, it involves the meiotic or the reduction division. The spermatocyte, it undergoes the first phase of meiotic division, that is the meiosis first, leading to the formation of haploid cells. These are called the secondary spermatocytes, and these are haploid in number, which are with the 23 chromosomes each. The secondary spermatocyte, it undergoes the second phase of meiotic division, that is the meiosis second, to produce the 4 haploid spermatids, and this spermatid, it is non-motile and non-functional. It gets transformed into a functional spermatozoa by the process of spermatogenesis. During this process of change, the spermatids remain held to each other and to the Sertoli cells by cytoplasmic bridges. The sperm heads remain attached to the Sertoli cells and their tails hanging in the lumen of the seminiferous tubules. During spermiogenesis, length of spermatid increases. The centrioles are rearranged as a primary and distal centrioles. And the mitochondria, it becomes spirally coiled, and acrosome, it is formed from the Golgi complex. So, this is the process of spermatogenesis. Then the next point, that is the structure of sperm. How the sperm is exactly seen. So, this is the microscopic. We cannot see with our naked eyes. So, here is the diagram of the sperm. This is the male gamete and it is a motile. Motile means movable and microscopic, elongated cell. It is divisible into the three parts: head, middle piece and the tail. First part, that is the head. The sperm's head, it is oval in shape and contains the haploid nucleus. Above the nucleus, there is a cap-like structure that is called the acrosome and it is formed from the Golgi body. This acrosome contains hydrolytic enzymes. These are hyaluronidase and proteolytic enzymes like zonalysins and corona penetrating enzymes. Second part, neck. It is a very short region having two centrioles, that is the proximal centriole and distal centriole. Middle piece. This middle piece, it has an axial filament which is surrounded by the 10 to 14 spiral turns of mitochondria. These mitochondria, they are producing the energy which is necessary for the movement of sperm. This is the very important function of this middle piece. In middle piece, there is a presence of the mitochondria. They are producing the energy which is required for the movement of the sperm. And the last part, that is the tail. The last part, that is the tail, it is a long, slender and tapering part containing the cytoplasm and fine thread axial filament. These axial filaments, there arises from the distal centriole and travels throughout the length of tail. It is partly surrounded by plasma membrane. The part without plasma membrane, it is called end piece. This is the structure of the sperm. Then next process, that is the oogenesis. This is also the same process like the spermatogenesis. Oogenesis, it is the process of formation of haploid female gamete, that is the egg or the ovum, from the diploid germinal epithelium. It involves the process of meiosis. So, like spermatogenesis, this oogenesis, this process, it is also divided into the three stages. First phase, that is the multiplication phase. Second phase, that is the growth phase, and the third phase, maturation. So, here is the diagram of this oogenesis or the division of this primary germinal cells into the ovum or the egg. First phase, multiplication phase. In this stage, the primary germinal cells, these are diploid in number of the ovary. undergoes the repeated mitotic division to form the millions of gamete mother cells. These gamete mother cells are also known as the oogonial cells. These are also diploid in number. This process, it is completed in the embryonic stage of human females. Then second phase, that is the growth phase. In this phase, some of the oogonia, they stop the division and begin to increase in size and form the primary oocytes. This is also diploid in number. In this phase, cellular organelles like endoplasmic reticulum, Golgi apparatus and mitochondria increase in number. These cellular organelles, for example, endoplasmic reticulum, Golgi apparatus and mitochondria, that increases in number. Then third phase, maturation. This oogenesis takes place in the ovaries. The process, it is initiated prior to the birth of female baby. The primary oocytes, which are diploid in number, enter the maturation phase, which includes the meiotic division, means meiosis first and meiosis second. The diploid primary oocyte undergoes the meiosis first to form the two haploid daughter cells. This division is peculiar in females as both the daughter cells are with haploid number of chromosome, that means the 23 chromosomes. But due to the unequal division of cytoplasm of the two daughter cells produced, one is a large cell, that is called the secondary oocyte, which is haploid in number, and another is a small cell called first polar body. This is also haploid in number. Normally, the first polar body does not enter meiosis second. The secondary oocyte, which is haploid in number, it proceeds meiosis second only up to the metaphase second. Its division is further stopped or arrested at this stage. The secondary oocyte is shed from the Graafian follicle and ovary. The restart and completion of meiosis second will happen only with the entry of the sperm. This last phase is usually completed into the ampulla of fallopian tube at the time of the fertilization only. In this division also, the two unequal daughter cells are formed. The large cell is ovum and the small cell, it is called the second polar body. The ovum so form functions as a female gamete and is ready for the fertilization. Then, after the process of oogenesis, there is a structure of secondary oocyte. This secondary oocyte or the unfertilized egg or ovum. In humans, unfertilized egg when ovulated, means when released from the ovary, is actually the secondary oocyte. It is non-cladoic. Cladoic means without shell. There is a no shell, and microlacithal. Microlacithal means yolk is present in very small quantity. Approximately, it is 0.1 millimeter in size. It is rounded, non-motile and haploid female gamete. This nucleus of the egg, it appears large and it is called germinal vesicle. Typical nucleus is formed at the time of the fertilization only. The cytoplasm of this egg, it is also called ooplasm, which is present in the ovum or the secondary oocyte. That's why it is called the ooplasm. This egg, it is surrounded by the various coverings. So, here is the diagram of this unfertilized egg or the ovum. This egg, it is surrounded by various membranes or the coverings. The egg membrane, it is called a vitelline membrane. It secretes the non-cellular glycoproteinous membrane, zona pellucida, on its outside. Adhering to the outer surface of zona pellucida are several radially elongated cells forming the corona radiata. These cells are derived from the innermost layer of granulosa cells. They are firmly held to the zona pellucida and to each other by hyaluronic acid, that is a mucopolysaccharide. Between the vitelline membrane and the zona pellucida, there is a presence of the fluid or this cavity or the space which is filled by a fluid, that is called a perivitelline space. The first polar body lies in this space. The egg shows polarity. The side having germinal vesicle and first polar body is called animal pole, while the side opposite to it is called vegetal pole. This is the structure of the secondary oocyte, egg or the ovum. Now, in this lecture, we discuss about this process of spermatogenesis and oogenesis. Next topic from this chapter, reproduction in lower and higher animals, we will discuss into my next video lecture. Till then, stay home, stay safe. With this, I am giving you a homework also. You have to complete the homework, and this homework it will check when your college will start. You have to write the answers of the questions into the notebook and keep these notebooks safely. That's it.